Hello students, how you all are? I hope you all are doing good, and the chapter we are going to start today is Microbes in Human Welfare, which will also be coming in the term 2. So your term 2 will be prepared with this chapter as well as the NEET. So without delaying, let's start the chapter. Yeah. So first of all, let's understand what are microbes. Microbes are microorganisms, right, which can be used in the welfare of humans, like bacteria, like fungi, like certain kind of viruses. Okay, so how these microorganisms are useful for humans? This is what the chapter is all about, microbes in human welfare. So there are several categories in the chapter that where microbes are used and how they can be used in the human welfare. So let's start. First of all, we will be dealing with microbes in household products. How usually in the everyday life the microbes are helpful for us. The question comes directly from this portion, right? You have to learn everything along with me, and then your chapter will be prepared. So let's start, beta. Curd. You know that the curd is formed with the help of the bacteria Lactobacillus. How? The Lactobacillus bacteria, it performs anaerobic respiration, and anaerobic respiration is the respiration which occurs in the absence of oxygen. And what kind of anaerobic respiration is seen in this bacteria? That is lactic acid fermentation. So it forms lactic acid, and that lactic acid, it precipitates the casein protein present in the milk, and this is how it makes the curd. Okay, so what Lactobacillus does, it coagulates the casein protein present in the milk. The casein protein present in the milk, and this is how milk is converted into curd. Okay, so you have to remember curd is formed by the bacteria Lactobacillus. Learned? Now second, cheese. Cheese, you have to remember there are several kinds of cheese. Okay, some of them are formed with the help of bacteria, and some of the quality of cheese, they are formed with the help of fungi. So you have to remember, beta, that Swiss cheese is formed with the help of bacteria, that is Propionibacterium, whereas the Roquefort cheese and the Camembert cheese, these are formed by fungi, and Penicillium roqueforti, Penicillium camemberti. Okay, so what kind of question comes from here? It will say, the question will say that Roquefort cheese is made by a bacteria. Would that be false or true? It would be false, because the Roquefort cheese, it is formed with the help of a fungi, and the name is Penicillium roqueforti. So you have to remember this, beta, that Swiss cheese is formed by a bacteria, Propionibacterium, whereas the Roquefort cheese and the Camembert cheese, they are formed by the fungi Penicillium. Direct question comes, okay? Then comes dosa and idli. Question of 2019. Dosa and idli, right? It is a notion that dosa and idli are usually made by yeast. No. This is because when the question came, most of the students, they answered yeast, that yeast is helpful in the formation of dosa and idli. But actually, when you ferment the solution, when you ferment the batter for dosa and idli, actually the fermentation is done by the bacteria Leuconostoc and Streptococcus. So this is important because it is given in NCERT, right? So dosa and idli, remember, beta, dosa and idli is formed with the help of bacteria, and the bacteria are Leuconostoc and Streptococcus. Then comes yogurt. You know what yogurt is, right? So yogurt is formed by the bacteria Lactobacillus or Streptococcus. So I hope this slide is learned. So learn along with me. The curd is formed by the Lactobacillus bacteria, Swiss cheese by bacteria Propionibacterium, Roquefort cheese and Camembert cheese by the fungi Penicillium, dosa and idli by bacteria, that is Leuconostoc and Streptococcus, and then Lactobacillus and Streptococcus, they are helpful in the formation of yogurt also. Let's move on. Now we know a very important organism, that is yeast. You know, yeast is a member of Ascomycetes now. It is a unicellular fungi, and it is used both in the formation of of bread in the bakeries, and it is also used in the formation of alcohol. So that is why yeast is also called as baker's yeast or brewer's yeast, right? So because yeast is helpful in the formation of bread, that is why it is called as baker's yeast. And you know that the bread is fluffy. There are pores which are present in the bread. How that fluffiness and porous nature of bread comes from? The yeast, it respires anaerobically and produces CO2. And because of that CO2, this question directly comes. Because of this CO2, which is a result of fermentation done by the bacteria, by the yeast, Yeast performs alcoholic fermentation. And what are the products of alcoholic fermentation? Ethanol and CO2. And that CO2, when it gets evolved, it creates pores and puffiness in the bread. Okay, remember this. Okay, then comes toddy drink. It is a very famous South Indian drink. Toddy drink is formed by the fermentation of sap of palm. Palm is a type of monocot. It's an angiospermic plant, and from the sap we collect the sap of the plant sap of the palm tree, and then it is fermented with the help of yeast, and that fermented drink is called as the toddy drink. Okay, then there are certain food products which are formed by the fermentation of various food material like soya bean, like fish, like bamboo. So when soya bean is fermented with the help of yeast, then these products are formed. Tempeh, tofu, sufu. This question can directly come. Tempeh, tofu, sufu. These are the fermented soya bean products. When soya bean is fermented with the help of yeast, then tempeh, tofu, sufu is formed. Okay? Let's move on further. Now microbes. Up till now, we have understood how microbes are useful in the household products. Now let's discuss how microbes are useful in the industrial products. So the first industry is the alcohol industry, right? You know that the yeast is used on a large scale in alcohol factories and plants where alcohol is made with the help of yeast, and that is why yeast is also called as brewer's yeast. And you know what is the scientific name of yeast? It is Saccharomyces. So you know that yeast performs alcoholic fermentation and it forms ethanol, C2H5OH, right? So now what is important here, if you remember respiration in plants, there is a line given that the maximum concentration of alcohol in naturally formed alcohol can be 13%, right? The naturally made alcohol are the alcohols which are non-distilled alcohols. Non-distilled alcohols, that means the naturally how much content of alcohol is present in the beverage. Okay, so generally wine and beer, these are naturally made alcohols, non-distilled alcohols, and the maximum concentration of generally the concentration of alcohol in beer is 3 to 6%, and that in wine is 9 to 12%. So what is important, that in non-distilled alcohols, the maximum concentration of alcohol is 13%. Why? Because after 13% of alcohol in the medium, yeast dies. Okay, so if you have to increase the content of alcohol in any alcoholic beverage, then you have to distill it. So in distilled alcohols, you can increase the alcohol content by distillation, like whiskey, brandy, rum, vodka, scotch, right? In all these alcohols, the amount of alcohol can vary from 50 to 90%. Okay, so what kind of question comes from here? What is non-distilled alcohol and what is what are the examples of distilled alcohols, and what is the maximum concentration of alcohol that can be present in the non-distilled alcohols. So you will say 13%. If the question asks the reason, that you will say after 13% of concentration of alcohol, yeast dies itself. Yeast dies. Okay, that is why. Now, yes, microbes are also useful in the formation of antibiotics. What are antibiotics? That means which are against the life of bacteria. Antibiotics are against the life of bacteria, but for us they are probiotics because they save our life. So for bacteria, they are against the life because they kill the bacteria. So discovery of antibiotics is considered as one of the greatest discoveries in the history of biology. And antibiotics were discovered by chance. It is a case of serendipity. What is the meaning of serendipity? When you discover something by chance. So what was, how the antibiotic was discovered? You must have heard the name of Alexander Fleming. Alexander Fleming was working on a bacteria, Staphylococcus. This is important. Alexander Fleming was working on the bacteria, Staphylococcus. So while he was working on the bacteria, he took a petri dish and poured his bacterial solution on the petri dish. But the petri dish in which he poured his bacterial solution, it was not cleared. It was not, it was contaminated with fungi. So after one day when he came back to his lab, he saw that all his bacteria were dead. So he thought, what happened that all the bacteria in this petri dish are dead? So he found out that the petri dish was contaminated with fungi. And what fungi was present in that petri dish? It was penicillium. So he discovered that the penicillium fungi secreted some chemical due to which the bacteria Staphylococcus died. And later on, he named that chemical as penicillin. So Alexander Fleming discovered penicillin and told that it kills the bacteria. Okay, but he did not explain the potential of penicillin, that it can be used as antibiotic on a large scale. He discovered first antibiotic penicillin, but the potential of penicillin to be used as antibiotic was explored later on by the scientists Chain and Florey. So if the question comes, who discovered penicillin, you will say Alexander Fleming. But if the question comes that the potential of penicillin as antibiotic was explored by, you will say Chain and Florey. Are you getting the point? So this is how antibiotics were discovered. Later on, the three scientists, Fleming, Chain and Florey, all three of them were given Nobel Prize in the year 1945. Okay. Now, what is important to learn here, that the first antibiotic was discovered from fungi, but most of the antibiotics are formed by bacteria. Most of the antibiotics are formed by bacteria itself. That means one antibiotic which you are extracting from one bacteria is used to kill the another bacteria. So the largest number of antibiotics available in the market, they are obtained from bacteria itself. And the largest genus of bacteria which is used for the formation of antibiotics is Streptomyces. This is very, very important. Streptomyces is a member of the bacterial class Actinomyces. So this question directly comes. Streptomyces is the bacteria from which the largest number of antibiotics are obtained. Some of the examples are erythromycin, streptomycin, chloramphenicol, neomycin, novamycin. There are several antibiotics which are obtained from the bacteria Streptomyces. Okay. So this was about the important. The first antibiotic was extracted from fungi, but most of the antibiotics are formed from bacteria, and the name of that bacteria, Streptomyces. Now, there are certain chemicals, there are certain organic acids which are also obtained from microbes and produced on a large scale in the industries. So let's see what kind of, this question is NEET favorite, is NEET's favorite since 2019, 20, 21, 18. These kind of questions, they come very often, that what kind of chemical and what kind of organic acid is obtained from what kind of organism. So this is very important, and this has come in NEET 2019 and 2018. Citric acid and gluconic acid are obtained from, your answer will be fungi, and the name of that fungi is Aspergillus niger. This is a member of Ascomycetes. Then acetic acid. Acetic acid is obtained from a bacteria, and the name of the bacteria is Acetobacter aceti. Then butyric acid is obtained from the bacteria Clostridium botulicum. That is why the bacteria Clostridium is also called as butyric acid bacteria. Then comes lactobacillus. Lactic acid is obtained from, these are very important chemicals, very. Look into my eyes and learn with me. Citric acid obtained from fungi, Aspergillus niger. Okay, lactic acid from bacteria, lactobacillus. Butyric acid from bacteria, Clostridium. And acetic acid from bacteria, Acetobacter aceti. Sometimes the question will come like this, that, acetic acid is obtained from a fungi. So this statement would be false. And if the question says, this citric acid is obtained from a bacteria, this statement will be false. Okay? Yes. Now there are certain enzymes also which are produced from certain microbes on a large scale in the industries, like lipases. Lipases are the enzymes which are used in detergents. When there is a cloth and there is an oily stain on it, when you wash it with detergent, the oily stain goes away. Why? Because lipases are present in the detergents. So this is very, very important, that where lipases are used. Lipases are used in the detergents, and they are obtained from the fungi. The enzymes, lipases, are obtained from fungi, and the name of the fungi are Candida and Geotrichum. Candida and Geotrichum. From them, lipases are obtained. Then, next important enzyme is amylases. Amylases are used to degrade starch. You know what are amylases, right? Amylases are the enzymes which degrade starch, and they are also obtained from fungi like Aspergillus and Rhizopus, and bacteria like Bacillus. So look into my eyes and learn with me. Lipases are the enzymes which are used in detergents to remove the oil stains, and they are obtained from bacteria? No. Lipases are obtained from fungi, Geotrichum and Candida. Then amylases are the enzymes which degrade starch, and they are obtained mainly from fungi, and the fungi is Aspergillus. You are hearing this name again and again, Aspergillus. That means this Aspergillus is important microbe, important fungi, right, in human welfare. Then comes important proteases and pectinases. These are also enzymes. Proteases and pectinases. Proteases break down proteins and pectinases break down pectins. You must have seen that when you make the juice at home, it is very, it is not very clear and not very smooth. But these bottled juices, they are so smooth and clear. So when the juices are bottled up, before that, the juices are treated with pectinases and proteases so that the juice becomes clear. So in bottled juices, pectinases and proteases are used to clear the juices. Okay, and they are obtained from the fungi, mainly from the fungi, Aspergillus, and Bacillus bacteria is also used. So again, Aspergillus, right? So amylases are also mainly obtained from Aspergillus. Proteases, pectinases, they are also majorly obtained from Aspergillus. So Aspergillus is pretty important fungi. Oh my God, this is favorite of boards and favorite of NEET. Streptokinase. This is a bioactive agent. Streptokinase is a bioactive agent which is used as a clot buster. What is the meaning of clot buster? If the clots are formed in the blood, they dissolve that clot. So what is the meaning of clot buster? That streptokinase is a bioactive agent which is helpful in removing or in dissolving the clots from the blood. And this is obtained from the bacteria Streptococcus. If the question says that streptokinase is obtained from fungi, it would be false. Streptokinase is obtained from Streptococcus. So you can easily learn this. Streptokinase, Streptococcus. And you know Streptococcus is a bacteria. Okay, this is important. Then after streptokinase comes, oh my God, again very important, cyclosporin A. You must have heard that when an organism undergo transplant surgery, suppose in an organism a foreign kidney is transplanted, right? There is a kidney transplant surgery. So the immune system of that person is suppressed so that it will not attack the foreign kidney and it will accept the foreign kidney. So for some period of time, the immune system of that particular person is suppressed, right, by using the bioactive agent cyclosporin A. So cyclosporin A is a immunosuppressive agent and it is used during organ transplants. It is obtained from the fungi Trichoderma. Cyclosporin A is obtained from fungi and the name of the fungi is Trichoderma. Then comes statins, again very important. What happens when certain people, they get heart attacks? So they are monitored. If one of the reasons of heart attack is the increase in the blood cholesterol level, which can block the arteries, right? So to reduce the cholesterol level in the blood, statins are used. Now the question comes, how they reduce the level of cholesterol in the blood? So your answer will be that statins inhibits or show competitive inhibition with the enzymes that are responsible for the synthesis of cholesterol. So when the statin will compete with the enzymes which forms the cholesterol, so these statins will not allow these enzymes to form the cholesterol. So if cholesterol will not be formed, so ultimately after a point of time, the level of cholesterol would be reduced in the blood. Okay, this question will or I'm not leaking the paper, this question can directly come into the boards, that how statins reduce the blood cholesterol level. The answer will be only in few words, and the answer will be that it reduces the blood cholesterol level by competitively inhibiting the enzymes which are responsible for blood cholesterol, for synthesis of cholesterol. And this question can directly come in NEET. Okay, so this is important. And they are obtained from, statins are obtained from fungi, and the name of the fungi is Monascus purpureus. This is a type of yeast. That means it is also a unicellular fungi. Okay, Monascus purpureus. So look into my eyes and learn with me. Streptokinase removes blood clot. Obtained from bacteria. Name Streptococcus. Cyclosporin A, immunosuppressive agent. Obtained from fungi. Name Trichoderma. Statins reduce blood cholesterol level. How? By showing competitive inhibition with the enzymes that are helpful in the synthesis of cholesterol, and obtained from fungi. Name Monascus purpureus. Now, how microbes are used in sewage treatment? What is sewage? A sewage is a polluted water body. A sewage is polluted water body, right? right, which has sewage in it. That means all the organic impurities and the inorganic impurities. So how we will treat the polluted water? So if I talk about the sewage treatment plant, so there are three, it is done in three parts: primary treatment, secondary treatment, and tertiary treatment. The process of primary treatment is a physical process. It's a physical process. That means you remove the physical impurities from the water. In the primary treatment, you just remove the physical impurities from the water. In the secondary treatment, the treatment is done with the help of microbes. The secondary treatment is done with the help of microbes. That means microbes are useful in removing the dirt, in removing the organic impurities from the water. And the tertiary treatment is the chemical. It's a physiochemical treatment in which you use certain chemicals and you use certain physical process like UV rays, reverse osmosis, and you use certain chemicals like chlorine gas, like perchlorate ions, to further purify the water. So primary treatment is a physical treatment. Secondary treatment is a biological treatment in which we use microbes, and tertiary treatment is a physiochemical treatment. Okay, now let's deal with them one by one. First of all, let's discuss what is primary treatment. I just told you that during primary treatment, only physical impurities are removed, right? So primary treatment is also done. Primary treatment is also done in two stages. One is sequential filtration. Another one is sedimentation. This question often comes. What are the two steps of physical treatment? First, sequential filtration. Second, sedimentation. Now, what is sequential filtration? During sequential filtration, the floating debris is removed. Whatever floating debris, what is floating debris? Like a piece of polythenes, lightweight physical impurities. Lightweight physical impurities which are floating on the surface of water, like plastic bottles, like polyethylenes. So, during sequential filtration, these kind of floating debris, floating physical impurities, they are removed. How it is used? Some gauge is used, right? Like you have the, you use the, it's a gauge type thing. It's a gauge type thing in which when the water moves through that gauge, let me show you the diagram. Yes. So here in the diagram, you can see this is a gauge like thing, and when water pass from this gauge, then the impurities, the physical impurities, they get stuck in it, and the water which comes out of it, it will not be having these floating impurities. Okay, so this is sequential filtration. This is sequential filtration. Okay, now what is sedimentation? The physical impurities which are heavy, like the pieces of stones, like soil, like sand, which is heavy. So that heavy physical impurities, they settle down, right? So these heavy physical impurities, they are removed by sedimentation. So what is done during primary treatment? Both the floating physical impurities and the heavy physical impurities, they are removed from the water. The floating debris is removed with the help of sequential filtration, and the heavy debris is removed with the help of sedimentation. And ultimately, the water you will get, it will not be having the physical impurities, both floating and heavy. So ultimately, this liquid you are seeing here, it is called as primary effluent, right? And this primary effluent is ready to go into secondary treatment. And what is, this question is very important. What is primary sludge? What is primary sludge. So primary sludge is nothing but all the solids, all the solid impurities which have settled down in the tank, which have settled down in the tank. This question comes, what is primary sludge? What you will say? Primary sludge are the heavy materials like stone, grit, pebbles, which have settled down in the tank. Okay, what is primary effluent? Primary effluent is the water which has no physical impurities and is ready to go into the secondary treatment. Okay, so now this primary effluent, this primary effluent will go into the secondary treatment. Now secondary treatment is called as the biological treatment, where we are going to use the microbes, the bacteria, right? So the primary effluent is passed to large aeration tanks. What happens now? The primary effluent which we have obtained from the primary treatment, this is passed on. This primary effluent, this water which has no physical impurities now, it is passed on to large tanks, and those large tanks are called as aeration tanks. Why these are called as aeration tanks? Because there is much amount of oxygen provided in these tanks. So what happened that this is the aeration tank, right? This is the aeration tank, and we provide a large amount of oxygen in the aeration tank, and you can see flocks in this aeration tank. Now what are flocks? Flocks are the mass of aerobic bacteria and fungal filaments which will be used in the purification of water. And how they will purify the water? They will remove the organic impurities from it. So this aeration tank has a large amount of aerobic bacteria and fungal filaments which will be helpful in removing the organic impurities, and the mass of the aerobic bacteria and fungal filament is called as flock. Now this primary effluent, the primary effluent we have obtained from primary treatment, is passed on to this aeration tank, and when the primary effluent is passed on to this aeration tank, the bacteria and the fungal filaments present in this aeration tank, they remove the organic impurities from the primary effluent, okay, and they reduces the BOD of water. The BOD of primary effluent was very high, but when the water is passed on to the aeration tank, the BOD of water gets reduced due to the aerobic bacteria. Now let's understand what is BOD. BOD, also called as biological oxygen demand, also called as biological oxygen demand or biochemical oxygen demand. If you take 1 liter of water, in which organic impurities are present, then the amount of oxygen, listen carefully, in 1 liter of water in which organic impurities are present, the amount of oxygen used by the aerobic bacteria, the amount of oxygen used by the aerobic bacteria to remove organic impurities from this water, that is called as biological oxygen demand. And obviously, the more the impure is the water, the more would be the demand of oxygen by the aerobic bacteria, because it will have to remove so much of impurities, right? So if water has higher BOD, that means it is dirty, and if water has low BOD, that means it is pure, right? So if the BOD is getting reduced, that means the water is getting purified. So we can say that BOD is inversely proportional to purity of water. If BOD is high, the purity of water will be less. If BOD is less, then the purity of water will be high. Okay, so dirty water has more BOD and clean water has less BOD. Are you getting my point? So what happens in the aeration tank? The aerobic bacteria, this question often comes, they confuse you. They say that in the secondary treatment, in the aeration tank, anaerobic bacteria is seen. False. During the secondary treatment in aeration tank, aerobic bacteria is seen. And what is the function of aerobic bacteria? To reduce the BOD of water. How? By removing the organic impurities from it. Okay. Now, effluent of aeration tank. See, what happens that the bacteria, it works rigorously to remove the organic impurities from the water. And then later on, when there is large amount of bacteria present in the aeration tank, then the bacteria and the fungal filaments, they settle at the bottom. And this is the water. The bacteria and the fungi, they settle at the bottom, and this is the secondary effluent, right? This is the water which has been treated now. So this is the secondary effluent, and the BOD, obviously the BOD of the secondary effluent will be very less because the aerobic bacteria has removed organic impurities from it. So now comes what is activated sludge. This again forms a very important question. Activated sludge are the flocks, are the masses of aerobic bacteria and fungal filaments which has settled down in the settling tanks. From aeration tank, this is transferred to the settling tank. And what happens in the settling tanks? The aerobic bacteria and the fungal filaments, they settle down at the bottom, and that is activated sludge. This is important. What was primary sludge? It was the heavy dirt, physical dirt. But what is activated sludge? They are flocks of aerobic bacteria and fungal filament which are settled down in the settling tank. Okay. Now this is the secondary effluent. That means the water which has undergone the secondary treatment, and now its BOD is reduced. Okay. Now, this is important that this activated sludge, some part of it is taken, only some portion of it is taken, and it is transferred back to the aeration tank, because in the aeration tank, the aerobic bacteria will be needed, right? So a part of the activated sludge is transferred to the aeration tank, for further use, right? For further use. But most of the part of the activated sludge is sent to anaerobic sludge digesters. Most of the part of this activated sludge is transferred to anaerobic sludge digesters. Now, what are these anaerobic sludge digesters? In the anaerobic sludge digesters, in the tank, which is called as anaerobic sludge digester, anaerobic bacteria is present. Anaerobic. And what this anaerobic bacteria does, it removes or it kills, it feeds on the aerobic bacteria and the flocks of fungi. Because we do not need that much amount of aerobic bacteria and fungi. Small amount of it was taken out and it was transferred again to the aeration tank, but the most of the part of it needs to be killed, because we do not want, that everywhere in the sewage treatment plant aerobic bacteria is there. So they are digested. The aerobic bacteria and the fungi is digested with the help of anaerobic bacteria. Okay. So in the anaerobic sludge digesters, which have anaerobic bacteria, it digests the aerobic bacteria and fungi. And because of this, what is this anaerobic bacteria here? This anaerobic bacteria here is methanogen. You must have heard the name of methanogen. It is a type of archaebacteria, right? So methanogen is a type of anaerobic bacteria, which is an archaebacteria. So this methanogen feeds on this bacteria and fungi, and along with it, when they are feeding on the aerobic bacteria and the fungi, they produce biogas as a byproduct. And what is the constitution of biogas? It has methane, hydrogen sulfide, and CO2. So see, this is so great that the water is also getting purified, and along with it, methane is also getting produced. So in anaerobic sludge digesters, the anaerobic bacteria, methanogen, when kills or digests the aerobic bacteria and fungi, as a byproduct, it forms biogas. So biogas is also formed during the sewage treatment. Now, the secondary effluent, remember the secondary effluent, the water which has undergone the biological treatment. Now this secondary effluent is directly released into the water bodies like river, because the BOD of the secondary effluent is reduced. Now this secondary effluent is directly released. This water is directly released into rivers, lakes, right? But if this secondary effluent has to be sent to the homes, right, for human consumptions, then it is not ready for human consumption. This secondary effluent is ready to be added to the rivers, but this secondary effluent is still not ready for human consumption. So if human consumption has to be there, then the secondary effluent has to undergo the third treatment, that is tertiary treatment. In tertiary treatment, which we use certain chemicals like zirconate ions, zirconium ions, perchlorate ions, chlorine gas, ozone gas, UV rays, technique of reverse osmosis, and then finally the water gets ready for the human consumption. So this is whole sewage treatment. Okay, what we have read up till now, the primary treatment, only the removal of physical impurities. First by sequential filtration and then by sedimentation. Then comes the secondary treatment, in which we use aeration tanks in which aerobic bacteria and flocks of fungal filaments are present. They remove organic impurities from the water and reduces the BOD of water. Then this is transferred to the settling tanks, where the aerobic bacteria and fungi gets settled at the bottom, and then this is called as activated sludge. This activated sludge is transferred to anaerobic sludge digesters, where methanogens, anaerobic bacteria, is present, which forms biogas by killing these bacteria. The secondary effluent is directly added to the water bodies, but if the secondary effluent has to be used by the humans, then it undergoes tertiary treatment. Okay, so this is a sewage treatment plant. Okay, so here you can see this is a container where the large pieces, the large floating pieces are removed. So what is this? What is happening here? Here sequential filtration is happening. Here sequential filtration is happening. Then after that, the heavy physical impurities like grit and sand, they settle down. This is called as sedimentation. Remember, this is called as sedimentation. This is sequential filtration, this is sedimentation. So this is the end of this is, and finally the sediments, they are, the sediments, they are given to the sediments have the outlet access. So this is the primary treatment. Okay, now this is the primary effluent. This is the primary effluent from which we have removed all the physical impurities. This is transferred to the aeration tank where we have aerobic bacteria, right? From there we have this settling tank where you can see the activated sludge here. This activated sludge, some part of it is gone back again to aeration tank, and most of the activated sludge is given to the sludge digesters where the anaerobic bacteria is present, and that is helpful in the formation of biogas. So this is a sewage treatment plant. Okay. Now the government, the Ministry of Environment and Forest, has initiated many plans for the treatment of water, like the Ganga Action Plan and the Yamuna Action Plan for the treatment of sewage. Now comes microbes in biogas production. We have just seen that during the sewage treatment plant, biogas is also produced as a bioproduct. But now we will specifically use the microbes for biogas production. So, what are the major steps which are involved in the biogas production? So, if I have to understand how actually the biogas is produced. So, the cow dung, right? The cow dung or the cattle dung, it is converted into a slurry. Some water and hay is added into the cow dung. So, a slurry is made out of it, a solution of the cow's dung. Then that solution is poured into a pit, and then that pit is covered. And when you cover the pit, you create anaerobic conditions in it because the oxygen supply is cut, right? So that pit has a slurry, has a solution of cow dung and hay and many other things. So, in those conditions, the anaerobic bacteria like methanogens, they produce methane in that slurry. So, what happens in that pit? First thing which happens in that pit is solubilization, then acidogenesis, and then methanogenesis. So, ultimately, biogas is produced in 3 steps: solubilization, acidogenesis, methanogenesis. So, first of all, let's understand the first step, that is solubilization. What is solubilization? The slurry, the solution of cow dung, right? It has complex organic materials in it. Those complex organic compounds like cellulose, fat and protein, they are, decomposed by the decomposer bacteria, and those complex compounds, they are converted into simpler compounds which are called as monomers. So what is solubilization? The conversion of complex organic compounds into simple organic compounds. Then these simple organic compounds, with the help of bacteria, fermentative bacteria, they are converted into organic acids like acetic acid, and this step is called as acidogenesis. And then ultimately, what happens? Ultimately, on these organic acids like acetic acid, which bacteria acts upon? Methanogen. Then ultimately, on these organic acids, methanogens, they act and they produce biogas. Okay, so first of all, solubilization, then acidogenesis, then on the organic acids, the methanogens act and they form biogas. Now the question directly comes, what is the constitution of biogas? Biogas, the chief constituent of biogas is methane gas, which is approximately 50 to 70% in biogas. Then after that, CO2 comes around 30 to 40%. After that, hydrogen sulfide comes 10%, and traces of nitrogen and hydrogen also. So this is very important. This direct comes in a question. What is the constitution of biogas? Okay. Now, the production of methane is done by special bacteria which are called as methanogens. And you know that methanogen is a type of archaebacteria. Methanogens are obligate anaerobes. Obligate anaerobes means they can only be anaerobic. They are found only and only in the places where oxygen is completely absent, like Methanococcus and Methanobacterium. And do you know one important thing about them? Methanogens are found, I have told you this in biological classification, that methanogens are also found in symbiotic association in the rumen, in the stomach of cattle, right? So this is a biogas plant. This is a biogas plant, and you can see that this is a slurry. This is a slurry which is formed of cow dung and water, right? So what happens that the methanogens, they form biogas, and then this biogas is collected later on. Okay. In India, the first biogas plant was installed, and it is promoted by two main organizations, and those organizations are IARI, that is Indian Agricultural Research Institute, and KVIC, that is Khadi Village Industries Commission. Direct question comes, which two main organizations are responsible for the promotion of biogas in India? Okay, so this is how microbes are helpful in the biogas production. Okay, now let's move on. Now let's talk about the biocontrol agents. What are biocontrol agents? These are some special organisms which can be used to control those organisms which are harmful for humans, or which are harmful for the productivity of crop plants which are used by the humans. Okay, so instead of using the harmful chemicals like insecticides, pesticides, why should I think we should use the natural means to control those organisms which can cause harm to the crop plants or the humans, right? So biocontrol agents are those agents which biologically controls their natural prey. So we have to exploit the predator-prey relationship here, right? How? Let's understand with examples. Like ladybird, right? Ladybird is the natural predator of aphids. Aphids are another type of insects, and ladybirds are also insects. So naturally, this ladybird can kill the aphids, which are also insects. So if we have any crop plant which is harmed by aphids, then instead of using the chemical insecticides, we can introduce ladybird in that crop land, and this ladybird will naturally remove aphids from there, right? So this is how the biological diversity will also be maintained, because this is a natural prey of the predator ladybird. Moreover, dragonflies are also a kind of insects which controls mosquitoes. Mosquitoes, they harm us, right? They cause malaria. So if we want to kill the mosquitoes, we can use dragonflies. So dragonflies is a biocontrol agent to kill mosquito. Ladybird is a biocontrol agent to kill aphids. Then comes Trichoderma. This is very, very important. Trichoderma is a fungi which is found in soil, right? In free living condition. That means it is not associated with, it is not in symbiotic association with any organism. It is found in free living conditions in the soil. And what it does, it protects the root of plants from several other pathogens present in the soil. There are several other pathogens like other harmful bacteria, other harmful fungi which can harm the roots of the plant. But this fungi naturally protects the roots of the plants from several other pathogens. So Trichoderma is a fungi which is found in free living conditions, not in symbiotic conditions, and they are found in root ecosystem which controls the pathogens from infecting the root. Okay, now comes Bacillus Thuringiensis. You must have heard this in biotechnology, that this bacteria, Bacillus Thuringiensis, it produces certain kind of spores, and those spores can control the insect population. So the spores of this bacteria are sold in market by the name of sporine. They are sold in the market with the name of sporine. And when you sprinkle these spores on the crop, they will control the butterfly caterpillars. These butterfly caterpillars, they harm the crop plant and they can be controlled by the use of Bacillus Thuringiensis bacteria. So Bacillus Thuringiensis is also a biocontrol agent. So here bacteria is a biocontrol agent. Then comes special viruses called as baculoviruses. And these baculoviruses are special viruses. They are also used as biocontrol agent. The genus of these bacteria, this is the common name and the scientific name, the genus of this virus is nucleopolyhedrovirus, that is NPV. Nucleopolyhedrovirus, NPV. These viruses, they are so useful to us. Why? Because they are species specific. That means these viruses, they kill only certain insects without harming any other organism. So they are very specific in doing their work. They do not kill a wide range of organisms. They harm only the insects. So that is why baculoviruses are narrow spectrum. They are not broad spectrum. They are narrow spectrum. They are species specific because they harm only certain species of insects, not all the organisms. Right? So they do not have any negative impact on humans, birds, fishes, because you know the bioinsecticides, they have so much of negative impact on the fishes in the aquatic body, on us. But these baculoviruses, they kill only insects and they do not have any harmful effect on us, on fishes and on birds. So they are very good biocontrol agents. And what you have to remember here, that they are species specific and narrow spectrum. Sometimes the statement will say that they are broad spectrum, false. Sometimes the question will say they are species non-specific, false. They are species specific. Okay. Now, bioherbicides. Bioherbicides are those chemicals which are produced by certain organisms which can be used to kill the weeds. What are weeds? Weeds are the herbaceous plants which are unwanted plants, which a farmer does not want in his cropland. So we use herbicides. Herbicides are the chemicals which have the side effects, various side effects. But instead of using the herbicides to kill those unwanted plants, we can use bioherbicides or weedicides. Bioherbicides are also called as weedicides. So prickly pear cactus, it's a type of cactus also called as Opuntia, a species of Opuntia. It naturally, prickly pear cactus naturally kills. Right, this prickly pear cactus, it is a problematic weed, right? It's a problematic weed. It's a weed which not good for the main crop. So if we introduce an insect, Cactoblastis, this insect will naturally kill this cactus because it feeds on the cactus. So if we want to remove weed, if we want to remove this cactus from our crop farm, so this insects can be used. Then Cercospora, it's a type of fungi which can be used to control another problematic weed, water hyacinth. You must have heard about it. This is also called as Eichhornia. It's a very problematic weed of water body. It's an aquatic weed. Water hyacinth, it's a aquatic weed. It's an unwanted plant in water bodies. It is also called as terror of Bengal. Right? So we can control this with the help of a fungi. Then we can also control the another type of, many type of weeds can be controlled, like milkweed vines. They are found, this milkweed vines, these are the weeds which are found in the citrus fruits orchards. Where the citrus fruits are grown, they are generally grown in between them and they can be removed with the help of fungi, Phytophthora, which produces the chemical divine. Okay, so these all are bioherbicides which are used to kill these unwanted weeds. Then comes the bioinsecticides. Certain plants secrete certain chemical which naturally kills the plant. Like you must have heard neem. Neem is used to whenever there were many mosquitoes in the village when we were used to be small kids. There were no all outs and good night coils. So what my nani or dadi used to do, they used to put some these leaves of neem around us. So the mosquitoes and houseflies, they did not used to bother us. So neem secretes a certain type of chemical as a directin, which is anti-mosquito. And derris is also a plant which secretes rotenone, which has the property to kill the insects. So these are bioinsecticides. Now there are certain crops which are also called as the smoother crops. Now what are smoother crops? Smoother crops are those crops which do not allow the growth of unwanted plants. They are so efficient that if those crops are present in the farmer's land, they will not allow the unwanted plants to grow. So if I am growing wheat in my farm, so in the next season, I should grow a smoother crop. And then after that season, I should again sow my wheat plant. So what will happen when I have done this crop rotation by smoother crop? The smoother crop secretes certain chemical in the soil which do not allow the growth of unwanted plant. So a farmer should often rotate its crop by smoother crops. And the example of smoother crops are barley, rye, sorghum, sunflower, alfalfa, soya bean, marigold. These all are smoother crops. So if a farmer does crop rotation with these crops, then in that farm there will be no problem of weeds. Okay. Now microbes as biofertilizers. You know what are fertilizers, right? Fertilizers are those substances, chemicals which are added to the soil, to increase the soil fertility. But now, if we do not want to use the chemical fertilizers, so there are certain microbes which are helpful in increasing the soil fertility. They are called as biofertilizers. Now, biofertilizer can be bacteria, can be cyanobacteria, or it can be fungi. All three of them, they are used to increase the soil fertility. So all three of them are biofertilizers. Now, if I talk about bacteria, right, you know that some of the bacteria are very good nitrogen fixers. They are called as diazotrophs. And what is the function of these bacteria? To convert N2 into ammonia. And they add ammonia to the soil, and that ammonia is taken up by the plants. So this is how the nitrogen fixing bacteria, they increase the soil fertility. And the examples are, this is very important, the examples of nitrogen fixing bacteria are Azotobacter, Azospirillum, Bijorhynchia. These are aerobic free living nitrogen fixing bacteria, right? Free living bacteria, free living. They are present, these bacteria which I have just named, Azotobacter, Azospirillum, Bijorhynchia. This is an important question. These three bacteria, they are present in the free living condition in the soil. That means they are not associated with any plant. They are present in the free living condition in the soil, and they convert N2 into ammonia. So they are aerobic bacteria. All three of them are aerobic bacteria. They are present in the free living condition in the soil and fixes nitrogen. Then Klebsiella, Clostridium, Bacillus, all three of them are anaerobic bacteria which are present in the soil in free living condition and fix nitrogen. So direct question comes. The question will say Azotobacter is a aerobic free living nitrogen fixer. True or false? True. If the question says Clostridium is a free living aerobic nitrogen fixer? False. Clostridium is free living anaerobic nitrogen fixer, right? So remember, Azotobacter, Azospirillum, Beijeringia, aerobic free living nitrogen fixers. Klebsiella, Clostridium, Bacillus, anaerobic free living nitrogen fixers. Then comes the Rhizobium. You must have heard the name Rhizobium and Frankia. These are the symbiotic nitrogen fixers. They fix the nitrogen when they are associated with certain plant. Rhizobium is found associated with legumes and Frankia is found associated with the plant Alnus. The plant Alnus, right? So this is important. Rhizobium, Frankia, they are symbiotic nitrogen fixers. Then comes the cyanobacteria, which is also called as the blue-green algae, right? Nostoc and Anabaena, these are the two cyanobacteria, the two blue-green algae, which are very important nitrogen fixers because they can fix the nitrogen both in the symbiotic condition and in the free living condition. So they are so efficient that they can fix the nitrogen in both conditions. This is a very hotspot question. Olocera and Tolipothrix, these are the cyanobacteria, the blue-green algae, which are found freely in the soil and they fixes the nitrogen. Okay, so Olocera and Tolipothrix are two examples of free living nitrogen fixing cyanobacteria, and they are often found in the rice or paddy fields. These two cyanobacteria or blue-green algae are often found in the rice fields and they fixes nitrogen in the free living condition. Okay. Then comes fungi. How fungi is helpful in increasing the fertility of soil? You know that fungi forms a very important association called as mycorrhiza, right? What is mycorrhiza? When roots of higher plants like gymnosperms and angiosperms, they gets combined with the fungi, that association, that symbiotic association is called as mycorrhiza. So this mycorrhiza is of two types, ectomycorrhiza and endomycorrhiza. If the fungi is present only in the surface of root, if the fungi is present only on the surface of root and helps the plant to efficiently absorb mineral elements from the soil, right? How they are helping the plant? How they are helping the plant, the fungi? It is helping the plant by efficiently absorbing the minerals like nitrogen, phosphorus, potassium from the soil. If the fungi is present only on the surface, this is ectomycorrhiza. And ectomycorrhiza is found in the Gymnosperm pinus. This is very, very important. And the fungi which gets associated with pinus is Boletus. So this question often comes. What is found in pinus? You will say ectomycorrhiza, ecto. And this association is so obligate that if Boletus is not present in the soil, then the seed of pinus will not germinate. Then comes endomycorrhiza. When the fungi, when the hyphae of the fungi enters into the root, into the cortical cells of the root, right? Then it forms the endomycorrhiza. Endomycorrhiza is also called as VAM. What is VAM? VAM is vesicular arbuscular mycorrhiza. When the fungal hyphae enters into the cortical cells, it forms arbuscules or tree-like projections inside the cortical cell of root. That is why endomycorrhiza is also called as vesicular arbuscular mycorrhiza. Okay? And this kind of mycorrhiza is found in the monocot like orchids. Orchids is a monocot, it's an angiospermic plant, and VAM, endomycorrhiza, is found in orchids. And this question is very, very important. What is the name of that fungi? What is the name of that fungi which most commonly forms endomycorrhiza? Your answer will be glomus. Your answer will be glomus, which is a member of the class Zygomycetes. So this is very, very important. Ectomycorrhiza is found in the plant Pinus. Endomycorrhiza is found in the plant Orchid. Ectomycorrhiza is formed by the fungi Boletus, and endomycorrhiza is formed by the fungi Glomus, which is the largest genus of fungi which forms VAM. What is VAM? It is endomycorrhiza. Okay, so I hope biofertilizers are clear. Biofertilizers are bacteria, cyanobacteria, fungi, right? So with this, our chapter is finished. I hope you have learned many things along with me, and wherever you find that you have to learn it more, just pause the lecture, learn it along with the lecture, and then move forward. And keep your NCERTs in front of you, and your chapter will be prepared. So all the best. Thank you.